We are recording. Yes. Okay. So today we're going to go to Chapter 3 module. Download the PowerPoint if you have not already. We're going to skip this handout because, well, obviously we're not in class and we're not going to do it. Here's your optional reading guide. And this is the Chapter 3 post-lecture quiz that you guys don't have to do because you are attending this lecture. You don't have to do it. I will mark you exempt from that one. All right, wait. Where did my PowerPoint go? What? Okay. Well, here it is. All right, let's get started. Biological molecules. These are molecules made by living things. Remember that vocabulary words are starred. Here's what we're going to discuss today: ATP, carbon compounds, and the 4 classes of biological molecules, which are carbohydrates, lipids, nucleic acids, and proteins. Let's talk about ATP. It's the energy currency of the cell, and it's called that because it is an energy carrying molecule. Whenever a bond is broken, energy is released, and whenever a bond is formed, energy is consumed. I'm going to try and turn my pointer into a laser pointer. There we go. All right, so this is ATP. It stands for adenosine triphosphate. The reason tri means 3. So we have 1, 2, 3 phosphate groups here. This is a simplified version of it. We have adenosine right here, and then our triphosphates are 3 phosphates. What happens is this is a really high energy bond right here between the last 2 phosphates. So when there's a process in the cell that needs energy, what happens is that the reaction will snag one of these passing ATP molecules and break that bond right there between the 2 phosphates. And energy is released into the surrounding environment. And then that allows the process or chemical reaction or whatever it is to consume that energy so that it can continue. And once that ATP is broken apart, we have this molecule over here, which is adenosine diphosphate. We've got 2 phosphates and another free phosphate molecule. And these guys, ADP and phosphate, will get put back together again in the mitochondria, which is why the mitochondria is called the powerhouse of the cell. It's because that is where ATP is made. And we'll go over that a little bit more in some more detail later. So we've got our ATP, we've got our ADP. ATP you can think of as a rechargeable battery. When it's in its triphosphate state, then it's full of energy. And when it's in its diphosphate state, it's a drained battery. But you can reuse a battery. It just has to get recharged. Let's watch this escape animation. Well, maybe if I can. Okay. Can you guys see this? I'm trying to show you a web page. Do you see a web page? Do you hear me? Is anybody listening?
Yes, ma'am.
Okay, cool. So
I'm a teacher just like you.
oh my
I
God,
teach things.
shut up. Okay. If you cannot hear the sound from the video, let me know.
Welcome to BioVid episode. When the bond is broken between 2 phosphate groups, that releases energy. Let's take a look at how exactly this happens in the cell. Here we have a process, and as you can see, it's in desperate need of some energy. Fortunately, there's ATP that comes along, and when that ATP comes to the process, it breaks the bond between 2 phosphate groups and donates 1 phosphate to that process or that protein or whatever it is that needs that energy. When this happens, the process gets the energy that it needs, and it can then do whatever task it needs to accomplish. Let's get rid of this process for now. And now you can see we are left with two phosphate groups, so it's no longer ATP, it's ADP, or adenosine diphosphate. If we want to use this molecule again, we need to recharge it as we would do for a rechargeable battery. In order to do this in the cell, we need a very special organelle called the mitochondria. ADP then goes to the mitochondria, and the ADP gets recharged by the mitochondria adding another phosphate on, and it's now ATP again. So whatever you're doing,
Were you guys able to hear the sound of that video? Ah, the chat. Yes. Yes, yes, yes. Okay. Excellent. Oh, we have someone else who joined. How exciting. All right, going back to the PowerPoint. Here's our first clicker question. I will start a multiple choice question. Remember, the room name is NWAC Bio, and I'm going to be waiting for 5 responses. Awesome! 100% chose A, which is the correct answer. Good job, guys. All right, that's all about ATP. Let's talk about carbon compounds. Carbon compounds are just molecules that contain carbon. There's 2 types of compounds, organic that contain carbon and inorganic that do not contain carbon. So you can see over here in this section, oh, I guess I need to turn this into a laser pointer again. No. Pointer options. Yes. Okay. So over here, these guys are inorganic, and you can tell because there is no carbon in them. Carbon, remember, is represented by the letter C. On this side, we have organic compounds. You can tell they're organic because there's some carbon in there. Carbon compounds are really important in biology. Carbon can form 4 bonds. It can bond with 4 things, 1, 2, 3, 4. Remember, every straight line represents a chemical bond. A hydrocarbon, this is a good vocabulary word, is a molecule made only of hydrogen and carbon atoms. So over here, this is a hydrocarbon. All we have is hydrogen and carbon making up the entire molecule. Over here, we have a hydrocarbon. Carbon atoms form what's called the backbone of the molecule. So we have the carbons in the middle here. They are the backbone. And this one has a ring shape. Stop it! Cats. This one has a ring shape for a backbone. Let's talk about some shorthand. This right here is actually the same molecule as this. It is depicting the same thing, and it's using some shorthand. So what we're going to do, is we assume that there's a carbon atom at each corner. So we have one carbon atom here, that's the first one, another there, another there, and another there. And then we're going to add hydrogens. Remember, carbon bonds to 4 things. So we add hydrogens in to wherever there's not enough carbon bonds. Let's do an example. This is shorthand. Let's make it into, well, not abbreviated. Let's make it into not shorthand. Here's how you do it. Assume a carbon at each point, fill in any empty bonds with hydrogen, and then keep doing that until each carbon has bonded with 4 things. So here, we're going to start by assuming a carbon at each point. So I've written in those carbons there. And now we're going to assume that we need some hydrogens here. So this carbon has bonded with 2 things already, and so we need 2 hydrogens. And the same with this carbon. It has 2 bonds, so it needs 2 more. Hydrogen is just the default that you fill in, and so on and so forth. So this is what this is abbreviated for. I don't want to say longhand. I don't know if that's even a word, but this is the shorthand. This is the not shorthand. There we go. Next clicker question. I tried to put a lot in to keep you guys engaged. All right. 75% chose D, which is the correct answer. 25% chose C. Carbon can bond with 4 things. Here's another clicker question. If you're watching the recording, you should be paying close attention to the clicker questions. They might be important for the quiz later. All right, 2 people answered this question. Oh, 3 people answered. Oh, wow. Okay. 33% said B, 33% said C, and 33% said D. Well, at least nobody chose A. That's good. Let's go back. Wait, what was the question? A molecule made only of hydrogen and carbon atoms is called a hydrocarbon. Hydro for hydrogen and carbon, well, obviously for carbon. So this is a hydrocarbon. This is a hydrocarbon. Let's try again. Well, this is also a hydrocarbon. Right. Yay, so much better. Everybody got C, hydrocarbon. Excellent. These things you do not need to memorize. I just wanted to show you these are some different ways that carbon skeletons can be different from one another. So we can have double bonds right here. They could be a ring structure. Here's the hydrocarbon that we were just looking at. They could be branched. And these are just a few of the many ways in which carbon skeletons can vary. Isomers, here's a vocabulary word. An isomer is a molecule that is this, isomers are 2 molecules with the same chemical formula, but their atoms are arranged differently. This is a great example, methamphetamine. One isomer is speed and the other isomer is a nasal decongestant. So take a look at this chemical formula. Remember, we were discussing that chemical formulas tell you the number and type of each atom within a molecule. So this molecule has 10 carbons, 15 hydrogens, and 1 nitrogen. And now let's look at the decongestant. 10 carbons, 15 hydrogens, 1 nitrogen. They are the same. What makes them different? Well, the arrangement of these atoms. This is a 2-dimensional representation of a 3-dimensional shape. So when we're drawing this, whenever you see this solid triangle, what that means is that whatever is at the end of the triangle, in this case CH3, is popping out of the screen towards you. And whenever you see this ladder-shaped triangle, what that means is that whatever is at the end of the triangle, in this case CH3, is pushed back. It's going back into the screen away from you. That is the only difference between these 2 isomers. Isn't that crazy? Speed or nasal decongestant. Tiny things can make a huge difference. Let's talk about functional groups. Again, you don't have to memorize these, but take a look at this picture over here. These are what's called different functional groups. Each one gives a specific function to the molecule that it is a part of. Because an organic compound's properties depend on the size and shape of its carbon backbone and the atoms attached to the backbone. Atoms may be part of functional groups. These guys give molecules specific chemical properties. So for example, OH, hydroxyl group, you often see those in alcohols. Carbonyl groups, C, double bonded to oxygen. You'll often see that in amino acids. Amino group, NH2, we also have those in amino acids. Phosphate group, this is often associated with either molecules that are polar or molecules that carry energy, such as in ATP. We have our 3 phosphate groups. Small changes can make a big difference. Here we've got 2 lions. One is male, one is female. Here's a little difference between them. Right here at this joint between this carbon circle and that carbon circle, we have a methyl group. And then over here, we have a carbon double bonded with an oxygen. Notice that here in the female version of the hormone, we're missing the methyl group. And over here, the oxygen is replaced with a hydroxyl group. Those are the only 2 differences between these 2 molecules. And just looking at the picture, like, look what a huge difference it makes. What are you doing? Excuse me. They know that I'm not paying attention to them, and so they're naughty. Let's talk about subunits. Think about Legos. When you have a bucket of Legos and you dump them out all out on the table and you're ready to make, I don't know, Darth Vader structure. What's that called? Death Star. A Death Star. You start with each individual Lego and then you put them together in different arrangements. Much the same way, carbon compounds have individual subunits that are put together in different ways to make the overall structure. A monomer is what we call a single unit. This is a monomer. A dimer is formed of two linked monomers. That's a dimer. A polymer is formed of many linked monomers. And once you have the overall structure, we call that a macromolecule. The word macro means large. So it's a very large polymer. This is an example. Glycogen is a macromolecule. And each of these little hexagons represents one monomer. So you can see how many monomers it takes to make a macromolecule. And this is just a part of the glycogen molecule. It's not the whole thing. Here's our next clicker question. All right. 67% said A, which is the correct answer. 33% said C. Recall, this is a monomer. A monomer is one unit, and a polymer is many monomers. Poly meaning many. Oh, well, I just answered this for you, so it should be real easy. Yes, you all chose C, which is the correct answer. A polymer is many linked monomers. All right, when you are studying for the test, you can fill this in. It's typable, so you'll be able to go into your PowerPoint and type on it. It's just a nice little review worksheet of all the things we're going to talk about today. So let's talk about the structures of carbon compounds. Monomers, here are two monomers. We have glucose and we have fructose. They are linked together through a chemical reaction called a dehydration reaction. Sometimes it's called dehydration synthesis. What this means is that water is a product. So think about the word dehydration. What does dehydration mean? It means you don't have enough water. Or when you dehydrate something, like my mother used to make her own, oh God, words escape me today. It's because I'm stuck at home, that's why. Beef jerky. Yes, she used to make her own beef jerky and she would put it in this thing called a dehydrator, and that would suck all the water out. Similarly, a dehydration reaction takes water out of the molecules, and therefore it's a product. So over here, we've got that hydrogen in yellow, and this oxygen and hydrogen also in yellow. Those are the ones that are going to join together to make the water product. And now these molecules have been joined through a dehydration reaction. The exact opposite is true of breaking them apart. That's called hydrolysis. Hydro meaning water, and the word lysis means to split or to break. So we are splitting this using water, hydrolysis. The water is added back into the molecules where it was in the first place. And now they are broken apart. And we're going to watch an animation. Ooh, there's something in the chat. What is it? Ooh, what's not working for you, Eli? Okay, cool. Excellent. So let's go over here to our modules. And at the bottom, we have our, that's not what I want, textbook animations. Let's go to chapter 3. You can always look these up on your own. Polymers.
Most biological molecules are very large and are built by assembling small molecules, or monomers, into long chains. The resulting molecules are called macromolecules, or polymers. A process of linking monomers, called dehydration synthesis, involves the removal of 2 hydrogen atoms and 1 oxygen atom to form water. One way this might happen is diagrammed here, where several generic monomers are shown with OH groups that could be used for linking. The animation illustrates the joining together of monomers by dehydration synthesis, catalyzed by a polymerase enzyme, and the reverse process, in which added water results in hydrolysis, catalyzed by a hydrolase enzyme.
All right. So here's our next clicker question. What kind of reaction is necessary to join 2 monomers? Yes! Everybody chose C, a dehydration reaction. Excellent. Now, the reverse reaction, which is not shown here, this is a dehydration reaction, so don't get confused. The reverse reaction, what is used to break apart monomers? Wow, you guys are amazing. These questions always get people, and 100% of you chose B, so good job. All right. Before we talk about the biological molecules, let's discuss some things that are common to all of them. They are large and complex. They are made only by living organisms. Remember, the prefix bio means life, so they are made by living organisms. They are organic, which means they contain carbon, and we often call them macromolecules. And there are 4 classes: carbohydrates, which are sugars, lipids, proteins, and nucleic acids. Lipids are fats and hormones. Carbohydrates have 4 calories per gram, lipids have 9 calories per gram, and proteins have 4 calories per gram. So you can tell by looking at this data here that the lipids carry the most energy, 9 calories per gram. Let's take a look at why that might be. Oh, also nucleic acids have 0 calories because we do not use them for energy. So triacylglycerol over here, this is a lipid. This has, notice all of those lines, all of those chemical bonds, and recall that energy is stored in chemical bonds. So we've got this huge molecule with a lot of energy stored in it. Let's take a look at glucose. This is glucose, a carbohydrate. And notice there are a lot fewer chemical bonds in that glucose, therefore it contains less energy. We are going to skip this because we're not in the classroom. All right, let's talk about carbohydrates. They are also called sugars. All of them act as an energy source and structural material. The monomer is called a monosaccharide, the dimer is called a disaccharide, and the polymer is called a polysaccharide. Here are some images of monosaccharides. We have glucose right here. That's the main energy source for cells. Fructose is another monosaccharide. That's in the fruits and it's sweetest. And galactose is another monosaccharide. It's the sugar found in milk. And there are, of course, other monosaccharides. These are just the ones that I want you to know about. Notice that they have the same chemical formula, but different arrangements of atoms that makes them isomers of one another. I found this on the internet. This person put C6H12O6 on their license plate, which is glucose, and that makes this car a sugar cube, which is fantastic. Let's just review here. The shorthand is a simplified structure. We're assuming a carbon at every point. In this image, we have assumed a carbon at every point still, but they've drawn out all of the other atoms, the hydrogens and the oxygens. And then in this one on the left, that's called the structural formula because it shows all of the carbons and hydrogens and oxygens. I just want you to know when you see these things, they all represent the same molecule. This is another way to represent them. So if you take these circular molecules, let's just look at this one, and you break it at any particular point and you stretch it out into a linear molecule, it looks like this. So this also is glucose, and then this is fructose. You don't have to memorize those, but I want you to know what they are when you see them. A disaccharide is the dimer of carbohydrates, and it consists of two joined monosaccharides. The three that I want you to know are sucrose, which is glucose plus fructose. That's sugar that you buy in the store, just normal sugar. Lactose, which is glucose plus galactose, and that is broken apart by the enzyme lactase. If you do not have lactase, you are lactose intolerant. You cannot break apart lactose into its monomers, glucose and galactose. And remember, monomers are always joined using a dehydration reaction, otherwise known as dehydration synthesis. Here's a picture of a dehydration reaction. We have two monomers. Here's the OH and the H highlighted that are going to come out of the reaction. So when those guys come together, the oxygen and the two hydrogens, they make water. The molecules have been dehydrated and now they are joined. Ooh.
Sometimes organisms link sugar molecules in pairs to form disaccharides. Here are several examples. Plants make sucrose by joining glucose and fructose. Sucrose circulates in plant sap, and we obtain it from sugarcane and sugar beets and use it as table sugar. Lactose is formed by joining galactose and glucose. Lactose is the disaccharide that gives milk its sweet taste. Maltose consists of two linked glucose molecules. Digestion of starch in a sprouting seed or in the intestine of an animal produces this disaccharide.
All right. This is another clicker question. Oh, hey, we've had this before. Think it might be important to know for the tests? Maybe so. Yes, you guys all remembered. 100% chose A. Very good. And the reaction that is the opposite. What reaction is used to separate monomers? Yay! Everyone chose C, hydrolysis. Excellent job, guys. All right, so we've talked about monosaccharides and disaccharides. Now let's talk about polysaccharides. This is the polymer of saccharides, of carbohydrates. This consists of many joined monosaccharides, and we have glycogen, starch, and cellulose. Those are the ones that I want you to know. Oops. I want you to know about these. They have different uses, but they are all made of many joined glucose molecules. The thing that is different between them is how the glucoses are put together. So here's a simplified version over here. We have starch, which is glucose monomers linked in the same orientation. You can see how the little stem is up at the top there. Cellulose, we have one glucose right side up and one upside down, and one right side up and one upside down, and so on and so forth. And in glycogen, there are branches that come off of the main stem here. All of these can be used for energy or structure, as you will see soon. This is just a picture, I think it's pretty, of potatoes. Here we've got some starch, some cellulose, some more cellulose and more starch, some glycogen. Just, it's all glucose monomers, all of it. Okay, that's enough about carbs. Now let's talk about proteins. Proteins are my favorite category because they are involved in nearly every function of life. 70% of a cell's dry weight is made of proteins. That can tell you how important they are in cells. They are the most diverse group of biological molecules. They can be enzymes, transport proteins, defense proteins, signal proteins, receptor proteins, contractile proteins, structural proteins, and storage proteins, and more. Basically, they do everything. This is a computer model of a protein over here. All proteins have a monomer called an amino acid. Their shape dictates their function. This is the most important feature of a protein, is its shape. If it does not have the right shape, it cannot do its job. It cannot have the right function. There are 20 amino acids, and depending on how they are put together and which ones you use, you can get millions and millions of proteins. This is the general structure of an amino acid. All amino acids have a central carbon. Remember, carbon can bond to 4 things. Here we've got the 4 things. The central carbon has a hydrogen. It has what's called an amino group, which is a functional group. It has a carboxyl group on the other side, which is a functional group. And then the only thing that varies between all these 20 amino acids is what is attached right here in this bond. We call that R. R stands for variable. I don't know why it's R random, maybe, I don't know. It's like the X in algebra. It can stand for anything. And here are the 20 possible R groups. Some of them have a positive charge, some of them have a negative charge, some of them are polar, and some are nonpolar. I want you to notice a couple of things. First of all, the general structure of the amino acid is the same in each of these cases, except for what's attached at the bottom of the carbon. That is what varies. Secondly, I want you to think about this like an alphabet. So in the English alphabet, we have 26 letters, and depending on which ones you use and how you arrange them, you can get millions of words. And in the same way, the protein alphabet has 20 letters, and depending on how you arrange them and how many you use, you can get millions and millions of proteins. So the monomer is an amino acid. The dimer is called a dipeptide. Take a look over here. We've got our 2 monomers. In this case, the amino acids are called glycine and alanine. Oh, look, here's our dehydration reaction. So we've got our oxygen and hydrogen and our hydrogen. Those are the highlighted ones that are going to join together to form water. Here's our water. And now we have a dipeptide. That is because this bond that joins amino acids is called a peptide bond. That's over there in the text. The polymer is called a polypeptide because named after the peptide bond. Poly means many. And those are always joined, remember, through a dehydration reaction and peptide bonds. Remember, the most important quality of a protein is its shape. When the polypeptide folds into a shape, then it can function. Then it is a finished functional protein. Well, part of that is blocked, but it should say protein, a long chain of amino acids, which is a polypeptide, folded into a 3D shape. There's our next clicker question. Okay, 67% chose D, which is the correct answer. 33% chose C. Monosaccharide is the monomer of a carbohydrate, not a protein. The monomer of a protein is an amino acid. Next clicker question. After a protein is made, what must happen for it to be able to function? Wow, we are split here. 33% chose A, 33% chose B, and 33% chose C. The correct answer here is C, because a shape is the most important feature of a protein. Remember, when a polypeptide folds into a three-dimensional shape, then it is a functional protein. Proteins can have 4 levels of structure: primary, secondary, tertiary, and quaternary. And I'm going to use this pipe cleaner to help demonstrate. So primary structure is just the sequence of amino acids. So just think of this as a polypeptide, a string of amino acids. Next question. What is a polypeptide? All right, 75% chose D, which is the correct answer. 25% chose B. A 3D overall shape is what an amino or is what a polypeptide needs in order to be a functional protein. But a polypeptide itself is just many linked amino acids. All right, so after the polypeptide, then the protein begins to fold on itself, and it can either fold into a spiral, this helix shape, or it can zigzag like this into what's called a pleated sheet. And it can do both of those if it wants. But in my protein, I've got a helix and a pleated sheet, just one of each. These are stabilized by hydrogen bonds. I'd like to draw your attention to, all right, no, there we go. I'd like to draw your attention to these dotted lines here between the sheets of between the amino acids. So these dotted lines here represent hydrogen bonds. Recall that hydrogen bonds are weak bonds, and the positioning of these hydrogen bonds determines whether the polypeptide will fold into a pleated sheet or an alpha helix. When you straighten your hair, what you're doing is applying high heat that breaks those hydrogen bonds and denaturizes the structure of your hair. So if your hair is curly, it normally has hydrogen bonds in a specific orientation, keeping that structure, keeping that alpha helix structure. But when you apply high heat, the protein denatures. It just becomes flat. That's why those hydrogen bonds are important. Here is our next question. Excellent. 100% chose B, which is the correct answer, a hydrogen bond. So that is, okay, wait, I denatured this. We got primary, we've got secondary structure. Excuse me. And now we come to the next level, which is tertiary structure. This is where the sheets and the helixes fold over on each other to make a three-dimensional shape. Now it's a functional protein at the tertiary level. How it folds depends on the interactions between the side groups of amino acids. So here we've got, I don't have the amino acids memorized, but I do know that this one is cysteine. So we've got cysteine right here on this side, CH2 bonded to S. And then on this side of the polypeptide, we have another cysteine, CH2 bonded to S. Now, those R groups, those S's, are going to interact with one another, and that causes the whole polypeptide to bend around in this bendy shape. In fact, the more cysteine you have in the keratin that is your hair, the curlier your hair is going to be because of these disulfide bridges. And that is also why a perm smells like sulfur. And quaternary structure is when two or more proteins come together to form a functional unit. So I've got my tertiary structure here, and if I were to join that up with another protein that has tertiary structure, that's quaternary structure. It's not always present. Some proteins can function just fine on their own. Sometimes they need a little help from another protein. So here's an example of a spray bottle. Spray bottles have all these parts, but none of the parts can actually function on their own. They are completed parts, but they can't function by themselves. They have to all work together. That's an example of quaternary structure. Individual proteins work together. Here's another example, is insulin. So here they're showing us the primary structure of insulin. We've got these two polypeptides. Remember, polypeptide is the primary structure. And then this is how it folds into an actual protein. We've got two polypeptides that came together, forming quaternary structure. We've been over this. I think this is the second clicker question that goes over this particular concept because it's important. Very important. All right. The correct answer is C. 75% chose C when it has folded into a three-dimensional shape. 25% chose B. A polypeptide, remember, is just a string of plain amino acids. Yes, you can sit here. A string of plain amino acids. And when it folds up into its three-dimensional shape, then it's functional. I just put this here to show you how little changes can make a huge difference in the overall meaning of a word. In the same way, tiny changes in one amino acid of a protein, or maybe two amino acids of a protein, that is going to make a huge change in the overall protein. Any ideas why? I'm going to open up a short answer question. So just think about it. If one of the amino acids is changed, if the primary structure is changed, why might that destroy the overall function? Excellent, because it can't fold perfectly and would be something else. Yes, it would be a different protein. In fact, it might be a non-functional protein. A protein is shaped for its specific. Oh my God, yes, you guys got it. Good job. All right, let's talk about enzymes. Enzymes are proteins or a group of proteins working together that catalyzes, which means speeds up, remember that from chapter 2, a chemical reaction. So here I've got this enzyme. This is, which one is this? This is a ribosome. Notice how it's shaped like a hand. It's perfectly shaped for the job that it's doing because this right here, this nucleic acid is running right through the middle of the protein. You are distracting me. Yes, I do love you. Okay. Right through the middle of the protein because this protein is perfectly shaped. If it were not shaped like that, it couldn't do its function. Enzymes participate in the chemical reaction, but they are not permanently changed, much like scissors. Where are my scissors? Here we go. Where's my paper? Oh, well, I have my bowl from lunch. Okay, so the scissors have a sharp part that's what actually does the job, and they can cut things like that. The scissors are doing the cutting, but they are not permanently changed. You can use them over and over again. Much the same way, enzymes participate in chemical reactions, but they are not permanently changed. They can be used over and over and over again. Enzymes always have tertiary or quaternary structural levels. A substrate is what the enzyme is acting on. In this case, the substrate is the bowl because that's what's being changed. And the active site is the place where the substrate fits. So in our example, that would be the blades. All right. Next question. Short answer question. What is an enzyme? Feel free to look back at your notes. Really, this is just to force you to look at the notes again. Yes. Very good, very good. A protein, a protein, a protein that is a catalyst. Yes. So the full answer is a protein that is a catalyst. A group of proteins that work together to speed up chemical reactions. Very good. It is a protein, yes, but not all proteins are enzymes. An enzyme is a protein that speeds up a chemical reaction. Let's go back. The point about shape is that the structure dictates the function. The shape of the substrate, in this case, this long rope here, and the enzyme, in this case, the hand, has to fit together perfectly. They must be complementary. Complementary just means that they fit together perfectly. It's like a lock and a key. If the lock and the key don't fit together, if their shapes are wrong, then nothing's going to happen. Some enzymes can work with multiple substrates, like scissors would be a good example of that. I can cut a lot of things with these scissors. Some of them only work with one substrate. Hello. We're going to watch this.
Most chemical reactions do not occur spontaneously in a cell. Instead, cells rely on proteins called enzymes to kickstart chemical reactions and speed them up, enabling cells to get the most out of the energy sources available to them. Enzymes have a unique way of kickstarting reactions. They work by binding to one or more specific molecules called reactants or substrates. Binding occurs at a special region on the enzyme called the active site. Once the substrates bind to the active site, they form what's called an enzyme-substrate complex. As the enzyme and substrates begin to react, some of the chemical bonds in the substrates begin to weaken, causing them to link together. Eventually, the chemical reactions at the active site lead to the formation of a different molecule. This is referred to as the product. Once the reaction has occurred, the product is released from the active site. The enzyme returns to its original state and is free to react again with another set of substrates.
All right, that's enzymes. So, to review, proteins lose their function if they lose their shape. In the process of what's called denaturation, the protein is unwound, it loses its structure, and it loses its function. What can do this? Really high heat, or if you freeze it, or an extreme change in pH. For example, if an enzyme which lives in your stomach is really acidic, it's really acidic in there. So the enzymes that are in your stomach are adapted to be in a really acidic environment. So if you take those enzymes and you put them in a really basic environment, an extreme change in pH, then they're going to unwind. They will denature. They will lose their shape and lose their function. Much like this beer bottle here has been smashed and it has lost its appropriate shape, and therefore it cannot hold beer. It cannot function. This hand has also been smashed. It cannot hold its correct shape, so it can't function. It can't hold things. If a protein loses its shape, it can no longer function. Here's our next clicker question. There's a little visual to act as a hint. Yes, yes, yes. Very good. It loses its shape, lost its shape, lost its shape. A protein's shape determines its function. Once it's lost its shape, it has no function. Perfect. Good job, guys. All right, here's our next clicker question. Awesome. 100% shows B, denatured. This is something we're going to be working on in lab 3. Okay. What determines the final shape of a protein? These 4 things. Which amino acids are used, the order that they're linked together, location of hydrogen bonds, and location of other bonds. Let's watch a textbook animation to watch this.
Hi, this is Kelly Hogan from the University of North Carolina. In this Visualizing the Concept video, we'll walk through how several layers of structure produce the functional shape of a protein. Let's get started. Life depends on proteins. These large molecules make up almost all the structures of a cell and mediate most all of its functions. Proteins are responsible for just about everything you do. For instance, enzymes are proteins that catalyze and regulate all the chemical reactions of your body. Other proteins, called transport proteins, move molecules into and out of your cells across the cell membrane. Some proteins act as signaling molecules that bind to protein receptors on cells and help to coordinate your body's activities. For example, the hormone insulin is a signal that causes cells to take up glucose. And the contractile proteins that make up your muscles let you dance. How can proteins perform these and so many other functions? What can you observe about the differences between these proteins? Right, they obviously differ in shape. A protein's function depends on its specific shape. Let's explore how that shape arises. A protein is made from amino acids joined by peptide bonds to form a polypeptide. The proteins of your body are composed of 20 different amino acids. You can see the different kinds indicated here by their 3-letter abbreviations. Let's take a closer look at the makeup of an individual amino acid. Each amino acid has an amino group, a carboxyl group, a hydrogen atom, and a variable group symbolized by R, and all are attached to a central carbon atom. The R groups of the various amino acids differ in their size and composition. An R group can be hydrophobic, meaning water-fearing, or hydrophilic, meaning water-loving. These properties help determine what shape a protein assumes. Note that leucine's R group is nonpolar and thus hydrophobic. Hydrophilic R groups may be polar, as shown here with serine, or even charged, as you can see in aspartic acid. So let's build a protein. The final functional shape of a protein is based on several superimposed levels of structure: primary, secondary, tertiary, and in many, but not all proteins, quaternary. Primary structure is the sequence of amino acids. This specific sequence is determined by instructions written in a cell's DNA. Added levels of structure, however, are required to turn a linear string of amino acids into the functional shape of a protein. There are 2 types of secondary structures. Portions of the chain may coil into an alpha helix. Other segments may fold back and forth, forming beta-pleated sheets. These secondary structures are held together by hydrogen bonds, shown here as dotted lines between hydrogen and oxygen atoms of the polypeptide backbone. You can see secondary structures compacted in the globular 3-dimensional shape of this polypeptide. The overall shape of a protein is called its tertiary structure. This shape results from interactions between R groups of the various amino acids. Hydrophobic R groups cluster in the center of a protein, out of contact with water. R groups that have positive or negative charges may form ionic bonds. Hydrogen bonds can connect polar R groups. and covalent bonds may form between sulfur-containing R groups. These disulfide bridges reinforce the shape of some proteins. You just learned that various types of interactions between the amino acids that make up a polypeptide chain produce the secondary and tertiary structures that determine a protein's shape. But can changes in the chemical or physical environment disrupt a protein's shape and thus its function? For example, could an increase in temperature or a change in acidity affect a protein? Yes. A physical or chemical change in the protein's environment may disrupt the chemical interactions responsible for secondary and tertiary structure, causing the protein to unravel. This process is called denaturation. Without its specific shape, a denatured protein loses its function. Many proteins consist of a single polypeptide, and thus have just primary, secondary, and tertiary structure. But some proteins have a quaternary structure, in which two or more polypeptide chains are aggregated into one functional protein, such as transthyretin, as seen here. In summary, a protein's functional shape results from 4 levels of structure: primary, secondary, tertiary, and sometimes quaternary.
Okay. Back to our PowerPoint. All right, lipids. That's all about proteins. Let's move on. Lipids are fats. They are called fatty acids, steroids, or cholesterol, depending on the type of lipid it is. All of them are at least partially hydrophobic. They're made of carbon, hydrogen, and oxygen, and they are special in that they do not form dimers or polymers. We'll go into that in a little more detail in a second. I found this great cartoon where one amoeba is talking to the fatty acid, and he says, "I'm not insulting you. You are a fatty acid." And the fatty acid is not happy about that. So a fatty acid, here's an example of a fatty acid right here. This, okay, come on. Pointer options, laser pointer. All right. There are 3 fatty acids in this molecule. Here is one of them. Here is another. And here is another. It even says 3 fatty acids there. And glycerol is a connector molecule. So a fatty acid is just a polar head and a hydrophobic hydrocarbon, remember that means only made of hydrogen and carbon, hydrocarbon tail. These guys can be linked together using what's called a connector molecule. In this case, it's glycerol. A fat, this is a fat, is just a large lipid made from the connector molecule glycerol and one or more fatty acids linked together. Fatty acid plus glycerol equals fat. This is triglyceride here. Remember the prefix tri means 3. We have 1, 2, 3 fatty acid tails. This is the fat found in steak. And there are monoglycerides and diglycerides. The term fat-free really just means there's no triglyceride in it, because when food makers were like, okay, let's see how we can sell more of our product, they were like, what if we call it fat free? And then they went to the government people who clearly hadn't paid attention in biology class, and they were like, okay guys, what requirements do we have to meet with our food to label it fat free? And the government people, again, not having gone to biology class, said, well, okay, as long as it doesn't have a triglyceride in it, you can say it's fat free. But monoglycerides and diglycerides, those are okay. So there's no such thing as fat free. It just means there's no triglyceride in it. Here is our next clicker question. What is a fat made of? All right, 67% chose D, which is the correct answer. Um, B, a polymer of glycerol. A polymer of glycerol doesn't actually exist. Polymer, remember, means many. A polymer of glycerol would be many glycerols. So that doesn't really exist. And remember, this is glycerol, the connector molecule. All right, I've got up here another fatty acid for you. We've got our polar head, we've got our long hydrocarbon tail. All of them have long hydrocarbon chain tails and polar heads, which means that one end is hydrophobic and one end is hydrophilic. And here is our very important note. Hydrophilic means polar, hydrophobic means nonpolar. All fatty acids contain carbon, hydrogen, and oxygen at least. Sometimes they have other elements. And they function for energy storage. Remember, look at all those chemical bonds, those lines that represent chemical bonds. So much energy can be stored in these molecules. Our next clicker question. Feel free to use your notes. Interesting, 50-50. Let's go back here. Hydrophilic, water-loving equals polar. So the terms hydrophilic and polar are interchangeable. That statement is true. Some more about fatty acids, saturated versus unsaturated. A saturated fat means there are as many hydrogens as can possibly be attached to that backbone. That means they're solid at room temperature. This is saturated right here. Notice how all those tails are straight. That allows them to pack in close together. All those fatty acids can pack tightly together, so they're solid at room temperature, like butter. Unsaturated fat is not saturated with hydrogens, so we've got some empty spaces here where there could be hydrogens, but there are not. And in order to compensate for that empty space, the molecule folds over to try and fill up the empty space. For this reason, unsaturated fatty acids have bends in their tails, like this one, so they cannot pack as closely together as the saturated fatty acids. So that makes them liquid at room temperature. This is my pet peeve right here. Okay, this stuff is not peanut butter, it is dessert. It is mashed up peanuts, but they have taken out the peanut oil because peanut oil is unsaturated, meaning it's liquid at room temperature. And people don't like to stir their peanut butter. So they took that out and then they put in an artificial oil called partially hydrogenated soybean oil. That means they started with soybeans and they artificially put all the hydrogens in there that they possibly could to make it saturated. This is, I don't know how they define that as natural, but no need to stir. There you go. No peanut oil. Again, they've taken out the peanut oil and they've put in this artificial partially hydrogenated soybean oil. This is actual peanut butter. This is just crushed up peanuts with some salt. You can see the peanut solids are on the bottom, and then on the top is the unsaturated peanut oil. So you do have to stir this, but it's actually peanuts. The most important fatty acid that we're going to talk about is a phospholipid. This is the main component of all membranes, and that is why it is so important, because it is the main component of all biological membranes. This is a phospholipid. We have a polar phosphate hydrophilic head and two nonpolar fatty acid hydrophobic tails. This is a shorthand. You just draw a little circle and then two tails. Here's our next clicker question. What is a phospholipid? Excellent. Only two people answered, but both of you got it right. The correct answer is A. All right, this is just an image of how when you take these phospholipids and just put them in water, they will automatically rearrange themselves into this sandwich shape. That means the polar hydrophilic heads face the water and they form a safe spot. They sandwich these hydrophobic tails between them so that the tails don't have to contact the water. This is what we call a phospholipid bilayer, meaning there's two layers, bi means two, of phospholipids. When I was in eighth grade, when I was teaching eighth grade, I asked my students to draw little cartoons about the biological molecules, and this kid chose a phospholipid. It's the phospholipid cooking show. So you see his polar head and his nonpolar hydrophobic tails, and he says, and then you add the water, and he puts the water in the bowl, but because his head is hydrophilic, he is sucked into the bowl and then ends his cooking show. I put this here because I want you guys to see how many membranes are in a cell. Remember that all membranes are made of a phospholipid bilayer. Here we've got our phospholipid bilayer. We have the cell membrane, which is the membrane surrounding the cell, keeping everything in. We have all these other organelles that are bounded by membranes, the Golgi apparatus, the secretory vesicles, the mitochondria, the rough endoplasmic reticulum, the smooth endoplasmic reticulum, the lysosome. You'll learn about those in the next chapter. But my point here is that there's a lot of membranes, so phospholipids are extremely important. Now, the cell membrane is different from the other membranes because it has things stuck in it. This is just an example, a picture of the cell membrane, the membrane that surrounds the cell. In here we have phospholipids. See our little phospholipids forming a bilayer. We have proteins, that's these big purple blobs are the proteins. We have carbohydrates, these green branches, those are carbohydrates. And we have cholesterol. Cholesterol is this yellow molecule that is stuck in between the phospholipids. And sometimes the cell will put cholesterol in the cell membrane to try and solidify it and tighten it up a little bit if it's getting too watery. Here's our next clicker question. Only 2 people listening. That's okay. Yes, you both chose D. That's the correct answer. They make up all membranes. Very good. Alcohol. I have an entire lecture devoted to the effects of alcohol, but I wanted to give you a little preview. So alcohol, when you drink too much of it, interferes with the pathway that makes phospholipids in your body. So instead of being saturated fatty acid tails, we have unsaturated, kinky fatty acid tails. So here we've got a nice, on the right, we've got a nice solid cell membrane, keeping things in that are supposed to be in, keeping things out that are supposed to be out. Whereas here, the cell membrane is more fluid. You can see there's some space between the phospholipids that make up the cell membrane. So things are going to be able to get out that shouldn't be able to get out, and get in that shouldn't be able to get in. And in order to compensate for this, the cell tries to stick some cholesterol into those empty spaces and like firm it up a little bit so we can keep the things inside that need to be inside and keep things outside that need to be outside. But basically, when you're getting drunk, you're ripping your cells apart. See, I know this and I still do it. And maybe you still will too. Anyway, all right. Now we're going to move on to steroids. Steroids are a kind of lipid that is including hormones and cholesterol. They are composed of carbon rings. As you can see here, we have this ring structure. And they're very common in animal cell membranes, and cholesterol is important for making other steroids. So right now in your body, you have steroids. All of your hormones are steroids. Cholesterol is a steroid. That's normal. The kind you don't want is anabolic steroids, which is the synthetic testosterone. All right, that's all about lipids. Now we are to the fourth class of biological molecules, nucleic acids. These are unique in that they're only used for genetic material, and they are the only class of biological molecule that is not used for either structure or energy. The monomer is called a nucleotide. Over here, we have a nucleotide. It's composed of a sugar molecule, a phosphate group, and one of 4 nitrogenous bases that can be adenine, guanine, thymine, or cytosine. It has no functional dimer. You either have individual nucleotides or you have a macromolecule, many, many nucleotides strung together. And the macromolecule is called DNA or RNA. Those are just the two types of nucleic acids. Here again, we have the monomer of a nucleic acid. This is a nucleotide. We've got our three components of the nucleotide, the phosphate group, the sugar, and the nitrogenous base, which can be one of four things. I want you to think of the nucleotides as letters in an alphabet. So we could have adenine, we could have guanine, we could have thymine, or we could have cytosine. And depending on which letters you use and how many you use and how you arrange them, you can get millions of different genes. Here is our next clicker question. All right. 100% chose D. Good job, guys. We are almost to the end here. Nucleic acids. There are many differences between DNA and RNA, but we're only going to go over a couple today. The rest we'll get to in chapter 10. DNA is a double helix. A helix is just like a spirally shape. So over here we have Khaleesi with her nucleic acid hair. DNA is a double helix, two spirals wound around each other. RNA is a single helix, just one spiral. So that's one major difference between DNA and RNA. They both contain instructions for making proteins, which means that they control the cell. Recall that 70% of a cell's dry weight is made of proteins. That just shows you how important proteins are to a cell. Here's the thing though. DNA controls the cell. The nucleus does not control the cell. The nucleus contains the DNA, but the DNA itself actually controls the cell. This is like saying your head controls your body. No, your brain is contained in your head, and your brain controls your body. Here is our next clicker question. I think this might be the last one. All right, excellent. Good job, guys. 100% chose D. All right, you can do this on your own if you want. Here's something that you can fill out by looking at the book if you want. And that is the end. I am going to stop the recording, but if you're still listening, I want you to hang around for an important announcement. Can we see your cat before we leave? Well, yes, you can, but I'm going to stop the recording first. Stop recording.